A very good evening to all of you. Yes, so I hope my voice is loud and clear. Can you please confirm in the chat box? Yes, I hope the PPT is visible. Are we good to begin with our today's session? So once again, a very good evening to you. Welcome to another session of CUET PG Prarambh Series. This is day 15 and topic for today's session will be apoptosis and cancer. And very interesting topic from Unit 8 from your CUET syllabus. Okay, great. So let us get started. Okay, thank you, Megha. Thank you, Nandan. Thank you, Jayas. Yes, so let us start with our today's session. You know, this topic is not only important from your examination point of view, but otherwise also. Like this is one of the fields where most of the researches they keep on happening. Okay, so and apart from that, this is included in the CSIR syllabus as well. Okay, so yes, what do we know about cancer? What is a cancer? Right, cancer definitely this can be termed as a disease, right, in which the cells almost anywhere in the body they may begin to divide in an uncontrolled manner. Now, if I ask you whether it will be correct to tell cancer as a genetic disease, as we could see here, it has been mentioned that it is a genetic disease, right? But in the terms that it can be traced to the alterations within specific genes, right? Any alteration, it happens in the gene which modifies the way the gene is producing a product, and that will make a cell to proliferate in an uncontrolled manner. Okay, but if you know, what do you know about the genetic disease or inherited disease? Like unlike other inherited diseases where what happens, in an inherited disease a defect will be present in the parent gene and that will be transmitted to the zygote, right? So then we term those diseases as the genetic diseases. Now in cancer, the genetic alterations mostly which lead to cancer, they arise in a somatic cell due to the genetic alterations in the DNA during the lifetime of the affected individual. Right? Did you get the difference? Means in cancer, we can expect the genetic alterations to happen in the DNA during the lifetime of the affected individual as well. But since that those alterations will be affecting the gene function, so in that way the cancer that can be considered as a genetic disease. Okay, because of these genetic changes, cancer cells become freed from many of the restraints to which normal cells they are subject. Okay, Megha, you want me to explain in both the languages, Hindi and English, for better understanding? Okay, sure, Megha, I'll be trying on that. Okay, fine. So I hope now you got a basic idea that what is cancer and why this is named as genetic disease, right? And the genetic alterations which may lead to cancer, you need to understand that they are not always to be present in the parent genes, but they could be acquired during the lifetime of the affected individual as well. Now, if we talk about the normal cell properties, right? Normal cell, it undergoes regulated division and differentiation, right? And apart from that, apoptosis, the programmed cell death, it does not wander away from tissue to start new colonies elsewhere in the body, right? I repeat, all these three properties are really important, right? You need to understand एक नॉर्मल सेल कैसे बिहेव करता है. वो एक डिवीजन, जब भी एक डिवीजन होती है तो उस पर रेगुलेशन होता है, राइट? लेकिन एक कैंसर सेल की डिवीजन पर से वो रेगुलेशन हट जाता है, राइट? So it divides in an unregulated or uncontrolled manner. Now, other point which is important here is apoptosis. So whenever a cell is detected with the DNA damages, it is found that the DNA damages, they are being piled up in the cell. So the cell is sent to apoptosis. It is sent for the programmed cell death mechanism, which we are going to learn in the next part of our session. Okay. So that is known as apoptosis. However, the cancer cells, they will not even obey this mechanism. Means they'll keep on dividing, and even if they are supposed to die, they'll not die. Okay. They will not undergo the programmed cell death mechanism. Okay. They'll show defiance to apoptosis as well, right? But we are talking about normal cell, and normal cell, it doesn't wander away from the tissue to start. Means जहां पर एक नॉर्मल सेल रहता है, जिस टिश्यू में एक नॉर्मल सेल रहता है, वो उस टिश्यू को छोड़ करके दूसरे टिश्यू में इन्वेड नहीं करता, right? So I hope now you have understood that what do we mean by these three properties. However, if we'll talk about the cancer cells, so these are defined by two heritable properties. They and their progeny reproduce in defiance of the normal restraints on the cell division. जैसे मैंने पहले बताया, कैंसर सेल जो नॉर्मल सेल के डिवीजन के प्रोसीजर या रेगुलेशन को ऑबे नहीं करता, right? And then it invades and colonizes territories normally reserved for other cells. So this property of the cancer cell is known as invasiveness. Invasiveness means cancer cells, they do have the potential to metastasize. Now that is a technical term which we use. Metastasize means they have this ability to invade into the other cells of the tissues, and there they will start growing up, okay, to form the secondary tumors which are known as metastasis. Okay, so the combination of both of these properties will in fact turn a normal cell into a cancerous cell. Are we clear till here? I hope the things are clear so far. All looks easy. Now further, talking about the key behaviors of the cancer cells in general. So they disregard the external and the internal signals that regulate the cell proliferation. Clear? Okay. जहां पर एक नॉर्मल सेल को जब सिग्नल आएगा कि उसको अपना सेल प्रोलिफरेशन या सेल डिवीजन को रोकना है, and that cell is going to obey those orders, right, which are coming in terms of the molecular signals. However, a cancer cell will not obey to them. They tend to avoid suicide by apoptosis, so they don't undergo the programmed cell death mechanism, that is apoptosis. They circumvent program limitations to proliferation, escaping replicative senescence. Now, this is an important term which you need to take down. Replicative senescence and avoiding differentiation. See what happens, the normal cells, they will not keep on growing indefinitely. Why? Because they will be showing this mechanism or phenomenon known as replicative senescence, right, where there will be a limit to their cell division, right, and definitely the cell division that will be associated with the replication even further. So in that case, it is bringing a limitation to the cell life. But again, the cancer cell, that they'll not be following replicative senescence, and because of that, they'll keep on dividing in an uncontrolled manner. They are genetically unstable, so yes, they are vulnerable to mutations and all. They escape from their home tissues, that is invasiveness. So invasiveness, that is one of the important characteristics of the cancer cell. They survive and proliferate in foreign cells. Okay, not only they invade, but they are very much able to grow, establish themselves at the secondary sites. So if you have any doubts so far, yes, you can post your doubts in the chat section. So if we talk about the major cellular events, right, which may make a cell cancerous. So here we have an overview, right? For making it simple, I have put it in this form. So overview of changes in the cells that cause cancer. So they resist cell death. So if the cell, they need to die, but the cells which will not be obeying those orders, right, which are coming in the form of the molecular signals, So definitely they may progress towards becoming cancerous. Sustain proliferating signaling, right? So they actually do not require any signals to induce them towards cell division, right? So they'll just keep on proliferating, and they will be evading the growth suppressors. So there are many factors, or there could be many other hormones, right, which may make a cell to stop with the further cell division. Okay, but again, the cancerous cells, they will not be following the growth suppressors, and therein they'll keep on showing continuous proliferation. They activate invasion and metastasis, enable replicative immortality. So this is definitely linked to replicative senescence. All right. So are you guys dropping your queries in the chat section, as I can't see any doubt popping up? So I hope this is all clear so far. Yes. Okay. Now, induced angiogenesis. Now, this is another important term which you need to know. Angiogenesis. See what happens. I have explained that the cancerous cells, they do have the tendency to invade and metastasize. Okay, metastasize means they will be establishing themselves at the secondary site. Now, to do that, they have to grow in size, right? Okay, thank you, Jaypreet Singh, for the confirmation. Right now, yes. So to grow in size, the cancerous cells, they need more blood supply. Why? Because else they will be deprived of oxygen. Okay, so they will be having the hypoxic environment, right? Means there will be lesser oxygen present, right? Now, to get more oxygen and to grow more in size, they induce angiogenesis. Angiogenesis means the development of the dense growth of blood capillaries or the lymphatic vessels. So they could be supported by good amount of oxygen supply, and hence they will be growing in size so that they can further metastasize. So these all terms, they are technical. I hope you are taking down of them. Angiogenesis, metastasize, and metastasis. So the secondary tumors, they will be forming up, right? Those are known as metastasis. Okay, right. Now, if I ask you one question here, what is the difference between cancer and tumor? So I'm going very slow so that all of you can follow what I'm explaining here. Right, Shivani, that is correct. Blood vessel formation. So I think you have pulled that answer for my previous question, angiogenesis. Now I have this question here. What is the difference between cancer and tumor? Do you think both are same? Yes. Are both of these terms same, cancer and tumor? See, tumor refers to the uncontrolled growth of the cells, right? Which may occur even in the solid tissues like organ, muscles, or bone. And tumor can be benign or it can be malignant. So both the types of uncontrolled cell growth which we'll find in these two forms, benign or malignant, that can be known as tumors. However, cancer, that is the name given to the malignant tumor, right? Means when it will acquire this tendency, when it will acquire this potential to metastasize, right, to break loose away from its original tissue site, and then establishing itself at the secondary site. And let me tell you that 90% of the deaths, they occur because of the malignant tumors. Definitely means the tumors which acquire the potential to metastasis. Okay, when normal cells lose control over their division, differentiation, and apoptosis, they become tumor cells or neoplasm. Now, what is neoplasm? Neoplasm, that is the term which refers to the new abnormal growth, right? Now, new abnormal growth, what do we mean by that? That cells, they are progressing towards showing the property where they will be acquiring more mass, okay, and they will proliferate in an uncontrolled manner. Okay, tumor is a result of an abnormal proliferation of the cells without differentiation and apoptosis. right? So I hope you have noted down these two terms, right? Benign and malignant. Now, moving further, now what is the difference between these two? Benign, as long as the neoplastic. So we have learned this term neoplasm on the previous slide. Neoplasm is any new abnormal growth. Okay. So as long as the neoplastic cells they remain clustered together in a single mass, the tumor is said to be benign. It contains cells that closely resemble the normal cell and they may function like the normal cells. Means they will be differentiated, they will be having a clear cut boundary around them. Okay, just like the normal cells do have. However, malignant tumor, right, means the cancer, this could be used for malignant tumor where the cells they will acquire the ability to invade the surrounding tissue, right? This is definitely the primary requirement of a tumor cell to turn malignant, right? Now apart from that, let me take help of this diagram to explain that what do we understand from benign and malignant. Okay, so here what we are looking, this is a normal duct, this is a normal glandular duct, and the yellow portion which you are finding, this is the basal lamina. Okay, these are the epithelial cells. Now the pink portions which you are looking here in the diagram, this is representing the benign tumor. Right, now we could see that the benign tumor cells, they are restricted to the boundaries, right? They are restricted to the basal lamina. They are not disintegrating it. Okay, so this is benign tumor. Whereas in the malignant tumor, now we could see, it has been shown by in a different color, the red cell or the red, yes, the red cells which you are finding here, this is representing the malignant tumors. Now they are disintegrating the basal lamina. So they'll disintegrate the integrity of the basal lamina, and in that way they will be acquiring the ability to invade the other tissues to form secondary tumors. Okay, so this is one of the important concept where you need to know about the differences between benign and malignant tumors. So the first thing definitely is about the metastasis. Okay, so the benign, they do not invade into the other tissues, where the malignant they do. Okay, benign, they will be growing in a contained area, they will be having an outer covering, right, which will be made up of fibrous connective tissue, and then they will be growing at a slow rate. Okay, they will be having more of the normal characteristics. Means जैसे एक नॉर्मल सेल बिहेव करता है, mostly they will be behaving like that only. They will be restricted in a boundary, right? They will be having the clear borders and boundaries. They will be growing at a slow rate, like I said. They will be differentiated again, just like the normal cells. And they can be cured, right? Unless and until they are having serious health complications on the individual. Sometimes they even don't need to be cured, but in case if they are turning harmful, so they can be cured surgically, right? By removing the localized tissue and all. But opposite to this malignant tumor, they do not have clear boundaries. They will be disintegrating the integrity of the boundaries, right? And thus they will acquire this property, the invasiveness. They will be growing at a faster rate, right, as compared to benign. And then they will not be, following the molecular signals, which will be making a normal cell to stop with the proliferation, right? And then the malignant tumors, they are very difficult to be cured, right? And this makes sense because they will be invading the other tissues, so removing the localized tissue surgically, that will not help. 90% of the deaths, they are caused because of the malignant tumors. Okay, so they may require the treatment using the combined treatment, wherein the surgery, the chemotherapy, the immunotherapy, the radiotherapy, so all of them, they would be required together to cure a malignant tumor. Although the benign tumor, they remain precancerous, however, they should be checked periodically, so that they shouldn't transformed into malignant tumors. Are we clear so far? Any doubt? Okay. If not, then let us move to our next slide. And here, let me show you that what are the steps of the metastasis. And to show metastasis, the cancerous cells, they need to acquire angiogenesis. So angiogenesis, like we had discussed earlier, the dense growth of the blood vessels, right? Now to metastasize, right, which is definitely one of the important characteristics of the cancerous cells, the malignant cell, they must survive and proliferate in an alien environment. Okay. Metastasis itself is a multi-step process. Now, cells, they have to break away from the primary tumor, they invade the local tissues and vessels, and they establish new cellular colonies at the distant sites. I'll be showing you all these steps, right, through a diagram in my upcoming slides, right? But now we need to understand that to show metastasis, the tumor cells, they will be firstly inducing the angiogenesis. Why? I had given you a hint on this earlier as well, because the environment inside the growing tumor, that will become hypoxic, right? Now to have more oxygen, they will be inducing the growth or the dense growth of the blood vessels. Okay. So, to grow large, a tumor must recruit an adequate blood supply. Thus, angiogenesis, the formation of the new blood vessels, is a necessity for the growth of both primary tumors and metastasis. So, till the tumors, they will be small, okay, like around 1 to 2 in size. So, they are avascular, right? They do not have this thick supply of the blood capillaries and all. Now, when it will be growing further, so it needs more blood supply or more lymphatic vessels. So therein it will start secreting the angiogenic factors, right? I repeat, it will start secreting the angiogenic factors that will make the blood capillaries, the surrounding blood capillaries and all, to grow towards the tumor cells. Okay. So here we can see this in the second step, right? In the first step, we could see that the primary tumor, they are still small in size, but now when they are growing up, so they want more blood capillaries to grow. So they are secreting the angiogenic factors. And in the third figure, we could see that now tumor, it has grown to a considerable size with then supply of the blood vessels, right, which will be supplying efficient amount, a sufficient amount of oxygen to them. Okay, now the blood supply that we could see here through these blood capillaries, it will be tortuous, it will be heterogeneous, it will be leaky, and have many branches with the dead ends. Yes, so are we clear with this? Jaypreet, Shivani, I hope this is clear. All these terms, angiogenesis, metastasis. Okay. All right, before we move to our next slide, so here let me share this information with you once again, how you can reach us, right? You can write to us at this address, or you can make a call at our toll-free number. And please join BioTecNika on the Telegram app, where you will be able to get all the updates regarding the upcoming sessions or classes on YouTube or elsewhere on the other platforms. Don't forget to post your feedbacks, testimonials. We always appreciate your feedback. Okay, how we can improvise with our classes or the other sessions, right? Which you would like to see on the YouTube, right? Download the BioTecNika app. Again, you will be able to get all the updates regarding everything which BioTecNika has to offer. Now let us move to our next slide, and here what we are looking, these are the steps in the process of metastasis. So like I had mentioned earlier, I'm going to show you that how the tumor cells, they show metastasis, right? So it actually occurs in steps. So here, to begin with, we can see this is normal epithelium. This is the basal lamina, right? Let me tell you that this is an illustration where the tumor is spreading from bladder to liver, right? Now here, again, we could see that this is a benign tumor, right, on the epithelial cell. And now, as part of the next step, the tumor cells, they are entering into the blood capillaries. Okay. but more commonly they enter into the lymphatic vessels, right? Ultimately the lymphatic vessels, they will drop the lymph in the bloodstream only, right? And lymphatic vessels being more wider, they may allow the clumps of the tumor cells to enter into them. Okay, so lymphatic vessels, they are wider, their walls, they are flimsy, so they will allow the clumps of the cell to enter into them. Anyways, the next step will be that these cells will be flowing, right, through the bloodstream or the lymph, right, as the case would be. Now, next, they will be ejecting out of these capillaries, the blood vessels or the lymphatic vessels, as we could see here. They are escaping from the blood vessels, and then they will reach the secondary site. They will establish themselves. They are very much able to survive there, okay, and then they will be growing to, forming thick growths, which are known as the secondary tumors or metastasis. Yes, simple, right? And we have already learned that to show metastasis, to perform metastasis, these cells, they should have extensive growth of blood supply. Okay, so therein they induce angiogenesis, right? Now here we have a cross section of colon adenocarcinoma, right, that has metastasized to the lung. So you could see, in the center of the tumor. Can you see this? This is where the cells, they are showing necrosis, right? Necrosis means the cells, they are dying, right? And they are overgrowing the blood supply. So that usually happens in the center of the tumor when the cells, they are overgrowing. So they will just overgrow over the blood supply. Okay, so they will start showing necrosis. So this kind of structure that would appear in the center of the tumor, right? So this is a tissue slice which shows well differentiated colorectal cancer cells forming cohesive glands in the lung. Okay, so I hope we are clear with this, right? Now let me tell you about the terms adenoma or adenocarcinoma, if you have heard of these terms. So let me tell you that every benign tumor, that will be having a corresponding term for the malignant tumor. Okay, like adenoma, that is a benign tumor, and adenocarcinoma, that is a malignant tumor. Okay, do we get this? Similarly, chondroma, right? Chondroma, that will be a benign tumor, and chondrosarcoma, that is a malignant tumor for cartilage. So like I told you that the benign tumors, they have the tendency to get transformed into the malignant tumors, and they should be checked periodically. Okay, because they might not cause you know the they might not cause harm to the body to that extent as a malignant tumor may, but still they may cause some health complications wherein they can compress the surrounding cells, right? That is known as mass effect. Or maybe if that is an adenoma means the tumor which is of glandular origin, राइट वो ट्यूमर जो कि ग्लैंडुलर ओरिजिन का है मीन्स वहां पर सेल्स एक ग्लैंड की तरह बिहेव करना शुरू कर देंगे सो there are chances that maybe there's overgrowth of some hormones and all. So this is the reason that even if that's a benign tumor, so that should be checked, right? So that it shouldn't get transformed into a malignant form. Now let us move to our next slide and let us know more about the basic properties of a cancer cell, right? Now, the first property which will differentiate a normal cell from the cancer cell will definitely be in their cell division pattern. Okay, now here what we are looking, if we are going to grow normal cells in a culture dish, so they will be growing in a monolayer, right? However, if we allow the cancer cells to grow in a culture dish, so they will start growing in clumps or multi layers. Okay, clumps or foci. Okay, right. Now, why it happens? Because they do not obey contact inhibition. Right. Now, this is another term which you need to take down. Another important property of the cancer cell, contact inhibition. So, what happens? See, when the normal cells, they will divide, and when they will get into contact with the neighboring cells or even to the bottom of the culture dish, they will stop dividing, right? So that is known as contact inhibition, right? However, the cancer cells, they will again not follow contact inhibition. Even when they come into contact with the neighboring cells or the bottom of the dish, so they will keep on dividing. So therein they will form multi-layers, or they will start showing clumps or the foci growth. Okay, so same thing we could see here in this diagram. The cells, they are dividing, and the cells, they become quiescent, means they will become inactive, right? When they will be able to form a single layer covering the entire bottom of the culture dish, they will not be growing further. This is about the normal cells. However, tumor cells, they will keep on proliferating even when they are getting into contact with the floor of the dish or with the neighboring cells. Okay, Saptala, you have asked that how those metastatic cancers will choose secondary site. Our secondary site can be any site or chosen site. That's a good question. Yes, they can grow anywhere. Okay, they are flowing through the bloodstream or the lymphatic vessels, and wherever they will be able to escape from the blood capillaries, they can show secondary growths. Do you get that? It's not like that this will happen very frequently. Far fewer than 1 in every 1,000 malignant tumor cell that enter the bloodstream will actually colonize a new tissue. Okay. So I hope that helps, that answers your query. Now let us move to our next slide and let us learn about the types of cancer. So both benign and malignant tumors, they are classified according to the types of cells from which they arise. Right? Most cancers, they fall into one of the three main groups: carcinomas, sarcomas, and leukemias or lymphomas. Again, you need to take down these terms. They could be directly asked in the exam. What are carcinomas, sarcomas, or leukemias? Right? Carcinomas include approximately 90% of the human cancers, and they are malignancies of epithelial cells. Like I was giving that example for adenoma. Okay, so adenoma, that is a tumor which is seen in the epithelial cells. Okay, epithelial cells of the glandular origin, like in the pituitary gland, the thyroid gland, the prostate gland. Okay, or even in the non-epithelial layers, which may start behaving like a gland, right? That was about the adenoma. However, carcinomas, if we talk particularly about carcinomas, so these are malignancies of epithelial cells. Now, see, 90% of the human cancers, these are carcinomas by nature. That makes sense because epithelial cells, they are more exposed to the DNA damages, right? DNA damage causing agents. Okay, this is the reason that they may acquire this property to proliferate in an uncontrolled manner very easily, and they will start developing carcinomas. Okay, now if we talk about sarcomas, they are rare in humans. They are solid tumors of connective tissues such as muscle, bone, cartilage, and fibrous tissue. Next, leukemias and lymphomas, which account for approximately 8% of human malignancies. They arise from the blood forming cells like hematopoietic cells and from cells of the immune system, respectively. Tumors are further classified according to the tissue of the origin, example lung or breast carcinomas, and the types of cells involved. Okay, so I hope this was easy. You just need to take down these terms. You need to memorize that which particular term is used for which category of cancer. Now, moving to our next slide, here we'll talk about carcinogens. So, carcinogens, that is the term which is used for the agents that initiate or promote tumor formation, right? Majorly, we can divide carcinogens into three classes: radiation, chemicals, biological agents. So, firstly, talking about the radiation, UV light, ionizing radiation such as gamma rays, alpha particles from radioactive decay that may, make a cell to undergo tumorigenesis. Okay. Next, we have chemical carcinogens. Now, it has been found that repeated exposure of an individual to the chemical carcinogens, it leads to mutations, and those mutations further will make a cell to progress towards carcinogenesis. Okay. And to give examples, aromatic hydrocarbon derivatives of them, such as aromatic amines, nitrosamines, alkylating agents such as mustard gas. A few of these carcinogens act directly on the target DNA, but more potent ones are those which are relatively inert chemically and become damaging only after they have been changed to a more reactive form by metabolic processes. Now, this point is important. What do we have here? See, I hope you have understood that carcinogens, chemical carcinogens, those will be the carcinogens of the chemical nature. Okay, like we have some examples here. Now, some of them, they will be targeting the DNA directly. However, the more potent ones, the more dangerous ones are those which are relatively inert, right? But they will become harmful only when they have been changed to a more reactive form by the metabolic processes which are occurring inside the body, right? Like by the action of cytochrome P450 oxidase, right? Now, actually, P450 oxidase that is supposed to convert the toxic chemicals into the harmless ones. However, in some cases, the chemicals, when they are subjected to the action of this enzyme, they will turn cancerous. All right. Okay. So, moving further, we have biological agents. So, biological agents like viruses and other infections, they contribute to a significant proportion of human cancers, right? 15% of the world cancers, means 15% in the world as a whole, viruses, they are responsible, right? Although it's not a large percentage, but still this is significant. Now, DNA and RNA tumor viruses, both of them, they could be involved. However, the main culprit, these are DNA viruses only. Okay. Viruses can either alter a cell's DNA directly or they act as tumor promoters. So, yes, viruses, they may have some genes which will be responsible for altering the cell division behavior. So, they will make the cell to undergo the cell division or cell growth in an abnormal way, right? That is the direct effect they can have. Or maybe they may be having some tumor inducing effects of the cell. Again, that will lead to the same result, but this is where the viruses can act directly as well as indirectly, right? Now, if I give you an example here, so liver cancer, right? If we talk about the liver cancer, that is very common in Africa or Southeast Asia, where the people, they are vulnerable to the infection of hepatitis B, right? So the people who will be having hepatitis B infection, so they'll show chronic hepatitis, so they are, prone to having liver cancer. Okay, so uh, that is an example where we are finding that viruses, they could be responsible for causing the cancer, right? And that has been actually proven, wherein the people who were having cancers, they showed the viruses inside their body. And this is the next example which I have given, that in the areas where the people are more vulnerable to having infection with these viruses like hepatitis B or hepatitis C, so they have shown to grow with liver cancers. Okay. In AIDS, the human immunodeficiency virus, HIV, it promotes the development of an otherwise rare cancer called Kaposi sarcoma. So again, you need to note down this name, right? Now, what do we have in this point? That if an individual gets infected with HIV, so there are chances for the individual to have a rare form of cancer, which is Kaposi sarcoma. So again, this is an indication that viral infection, that is stimulating the possibility for an individual to have the cancer. Okay, Kaposi sarcoma, that is a type of cancer that can form masses in the skin, lymph nodes, or other organs. Last, infection of the stomach with the bacterium Helicobacter pylori, which causes ulcers. It appears to be a major cause of stomach cancer. So not only viruses, but bacteria and parasite, they have also been shown to increase with the chances of an individual to have cancer in some cases. Now, this is a slide where you will find an idea over the carcinogens, right? I was talking about that how some carcinogens or how some chemicals actually they could be turned or could be transformed by the enzyme which are present inside the body only, right? Into more potent carcinogens. Okay, like aflatoxin, that's a carcinogen produced by moles. It is found in peanuts and grains when they are stored under humid conditions. Then benzo, alpha pyrene, dimethyl nitrosamine, and nickel carbonyl. These are carcinogens. These are found in tobacco smoke or coal tar. Right? So again, this part is important. Please note down these names and let me know if you have any doubt till here. Now, this is a table which is going to give you an information that what all viruses, right? They could be known as tumor viruses. Okay, hepatitis B virus, like I had given you this example. The infection by hepatitis B virus that may lead to chronic hepatitis and that may further cause the liver cells to develop cancer. Okay, similarly, we have some other examples as well. So this table will be helpful to know that what all viruses they can cause tumors. All right. Now let us talk about the genetics of cancer. This is really important, right? Genetics of cancer. So now here you need to know a term, cancer critical genes. Shall I write down here? Cancer critical genes. So these are those genes which have a tendency to show alterations or modifications, right, and thus leading to cancers. So more than 100 critical genes they exist, right, and here these critical genes they can further be seen in by dividing them into two categories. Okay, the first category, which includes the proto-oncogenes, right? Now they acquire or they show the gain of function, right? Wherein when they will be acquiring some function, so that will impart these genes the property wherein they will be leading to those changes causing the cancers. Okay, now to explain this, let me take help from this diagram. Now this is a normal cell, right? And what do we have here? That we have two copies of a gene, and all we could see that both the copies they are in the good state, right? They are not causing, they are not a reason for a cell to show the cancer properties. However, because of a mutation event, when we could see that one of the gene that has been modified. So these solid boxes, which you can find in the diagram, the red boxes, they are indicating the mutations. Okay, now one of the gene, it has acquired the gain of function, and now this proto-oncogene that will be known to be converted into the oncogene, which has the property of causing cancer, right? So all in all, we need to understand that acquiring a single copy of mutation for proto-oncogene will convert them to the cancer causing oncogenes. So activating mutation enables oncogene to promote cell transformation. Right, now we will, if we'll talk about the second category of the cancer critical genes, that is tumor suppressor gene. So they will be showing loss of function when they will be a reason for the cancer. So again, we'll be starting with the normal cell, and here when the mutational event is occurring, we could see that the mutation has occurred again in one of the copies of the genes, and here we could see that the hollow box has been shown. Okay, hollow box means the loss of function, and the solid box was about the gain of the function, right? This is one thing. Now these are tumor suppressor genes which are associated with the loss of function. So whenever they will be the reason for causing cancer, they will lose some function. And the other thing which is important here is that In case of tumor suppressor gene, the loss of only one copy of the gene will not be sufficient. Okay, that means we can say that they will be behaving in a recessive manner. So here you could see when both the copies of the genes, they have acquired the mutation, then only the cancer causing properties they will develop. Are we clear with this? So this is really important. Let me know if you have any doubt on this. I quickly repeat what I have explained here. We have two categories into which we can keep the cancer critical genes, the proto-oncogenes and tumor suppressor genes, right? Now the cancer causing or the active form of the proto-oncogenes, they are known as oncogenes, right? They behave in a dominant manner wherein the gain of function by one of the copies of the gene that will be showing the cancer properties. However, the later, the tumor suppressor genes, they require both the copies of the gene to lose the function if they will be causing cancer further. So these are tumor suppressor and the former are the oncogenes. Okay, now further let us move to the next slide. So here we have another diagram which will help you to understand this concept in a better way. So what we are here, we are having here, this is the normal cell growth, and this is where you can see the mutated tumor suppressor gene, right? But still it will be growing in a normal manner unless and until the second gene that will also acquire the mutation. Then only it will lose the control over the growth and it will turn cancerous. But in case of proto-oncogenes, acquiring a single mutation means acquiring mutation in one of the two copies of the gene that will make this oncogene to show the proliferative properties which will go out of control. Yes. Now let us take an example wherein this will help you to understand that how tumor suppressor genes they may lead to cancer. Okay, this is a very common example we can take, retinoblastoma. In fact, this was the first tumor suppressor gene to be discovered, right? Now, what happens retinoblastoma? I hope you are getting an idea from the term itself. This is a cancer or a tumor of retina. Okay, and the symptoms they appear in the early childhood. So the children they show the tumors in the retina in the eye. Now here we can have two types of retinoblastoma, right? The first one is hereditary or familial, and the second one is sporadic, right? Or non-familial. This is also known as non-familial. What happens in the hereditary or familial retinoblastoma? So as you could see here, a child is inheriting a normal RB plus allele, right, from one parent and a mutant RB allele from the other parent. So यहाँ पे क्या है, दो copies हैं, right? RB, as you know that there exist two copies of a gene known as alleles. So one of them it is normal and the other one is defective. Okay, so this is what do we have in a somatic retinal cell. Now, if a mutation occurs, right, during the lifetime of this of an individual or during the growth when the retina cells they are growing, right? If any mutation happens and the second copy of the gene that also gets mutated, okay. So herein now we could see that this is a homozygous cell and it will give rise to tumors in retina, right? So this is an example of tumor suppressor gene, right? Till one of the copy that was normal, right? This was behaving in a normal way, right? The cells they were all normal only. However, if the mutation that happens in the other copy as well, so that will give rise to tumors in retina. Okay, so the children who inherit a mutated copy from the parent, definitely they will be at more risk. And when the other copy that gets mutated, so they may develop bilateral retinoblastoma, means both the eyes they could get defective, they can have tumors. As we could see that every somatic cell will be having both the both the alleles in the defective form. Not only this, since every somatic cell will be having the defective copies, so there are chances for the development of other types of tumors as well. Okay, now let us look at the other case. This is sporadic retinoblastoma. So here what happens, both the copies they are normal, right? So this is somatic retinal cell. Both the copies, they are in the normal form only, right? Now, maybe uh, first mutation it has occurred, and because of that, one of the allele it gets defected, right? And then during the lifetime, the other mutational event that also occurs, so making the other copy of the gene to turn defective as well. So, as an effect of two separate mutations, we could see that both the alleles they have been transformed into their defective forms. So, this is again a homozygous cell, right, with mutations in both the alleles, and this will give rise to tumors in retina. Now, this is known as sporadic or non familial because the individual has not inherited the defective copy. However, he has acquired both the mutations during the lifetime. Hmm. Okay. But all in all, I hope we have understood that when the mutations will occur in both the copies of the gene, then only the tumor that will appear. Okay, now this table is very important, right? This is going to give you a summary that what all genes they could be responsible for causing cancers, right? And in what ways, how they are different from each other. So we have seen how proto-oncogenes they may contribute, right? So if we talk about the normal functions of these genes, they help in promoting cell survival or proliferation. Okay, and these are the examples of the products being produced by these genes, like anti-apoptotic protein, components of signaling, signal transduction pathway, right, that results in proliferation. So all in all, they have a common target, means they are helping the cell with proliferation. So now we can understand that if in case they get some sort of mutation where they acquire gain of mutation, gain of function, right, मतलब उनका फंक्शन और भी बढ़ जाए, राइट? तो उस लेवल पर जा करके अगर उनका फंक्शन प्रोलिफरेशन को हेल्प करने लगा, सो दीज genes they will turn into oncogenes, right? So mutations, they are genetically dominant. We need to understand that they will be behaving in a dominant manner, as we have learned through the previous diagrams. Okay, talking about the second category, tumor suppressor genes. They inhibit the cell survival or proliferation. Now, talking about the normal function of these genes, right? How they help with the regular or the cellular, the fundamental cellular mechanisms, right? How they contribute? They inhibit the cell survival or proliferation, just the way opposite to the proto-oncogene, right? So apoptosis promoting proteins. So definitely the proteins which will be promoting the apoptosis, they will be making a cell to undergo apoptosis, right? Or the checkpoint uh, controllers, right? So what are checkpoints? Again, they check the cell for the proliferation or for the division. Okay, सो दीज ऑल आर द ट्यूमर सप्रेसर जीन. सो जस्ट इमेजिन, आप इमेजिन करो, अगर एक जीन एक ऐसे प्रोडक्ट प्रोड्यूस कर रहा है जो सेल को डिवाइड करने से रोकने में हेल्प करता है, राइट? जब चाहिए कि एक सेल को डिवीजन से रोका जाए तो ट्यूमर सप्रेसर जीन्स के प्रोडक्ट्स वहां पर हेल्प करते हैं. लेकिन अगर ट्यूमर सप्रेसर जीन का फंक्शन में लॉस हो जाए, राइट? लॉस ऑफ फंक्शन हैपन. सो लॉस ऑफ फंक्शन म्यूटेशंस विल अलाउ अनरेगुलेटेड सेल प्रोलिफरेशन एंड सर्वाइवल, राइट? सो आई होप यू हैव अंडरस्टूड. प्रोटो ऑन्कोजीन्स को गेन ऑफ फंक्शन मिलेगा, ट्यूमर सप्रेसर जीन्स लॉस ऑफ फंक्शन दिखाएंगे. अगर इन दोनों में से कोई भी होता है तो सेल विल शो अनकंट्रोल्ड प्रोलिफरेशन. ओके, the next category we have is the genome maintenance genes. Okay, so normal function of the genes is repair or prevent DNA damage, right? So the genes which are involved in the DNA repair mechanisms and all. Okay, so again, if they show loss of function, so what will happen? The DNA repair proteins, enzyme, they will not be produced. Okay, and that will again lead to cancers. Yes, okay, Nahid, Fatima, good to see that you have understood. So I hope Jaypreet, Saptala, and the other people who are here, I hope all this part is clear so far. What all genes are involved. I hope you have understood the genetics of cancer. Okay. what are cancer critical genes, what are proto-oncogenes, what are oncogenes, what are tumor suppressor genes, and how they can contribute to cancers. Now, prominent among the classes of proteins encoded by tumor suppressor genes are, see, now this is something which we are going to learn in detail when maybe we will be preparing for CSIR, okay, because this is a level higher. Here you can just learn the examples, means you can just have a look over the names of those genes, right? The products of these genes, they, they are how they can be involved in causing those changes which will be making the cell to progress towards being a cancerous cell, okay, towards tumorigenesis or carcinogenesis. So here, to give you some examples, intercellular proteins that regulate or inhibit entry into the cell cycle, P60 and RB. So we have already learned the example for RB. I had told you that that is an example for tumor suppressor gene. Receptors or signal transducers for secreted hormones or developmental signals that inhibit the cell proliferation. Remember, the effect of the tumor suppressor genes, it is a way opposite to the oncogenes, right? They are known to inhibit or to regulate the cell division. Checkpoint pathway proteins that arrest the cell cycle if DNA is damaged. So P53, that is another very important name. So I hope you are taking down the names. P16, RB, then the receptors, then P53. Okay, so they all will be acting as the tumor suppressor genes. Proteins that promote apoptosis, enzymes that participate in DNA repair. So remember, loss of function for any of the genes which we have learned here on this slide, that will lead to cancer. Yes, Jason. This will be the last session for your CUET PG Prarambh series. Okay, so I hope you enjoyed all the sessions. You found all the sessions informative. Okay, now let us move to our next slide. Let me give you an idea that how the proto-oncogene, it could get converted to oncogene. This is really interesting. Okay, see, this is a proto-oncogene, and I'm sure you must be having an idea that the coding part that has a regulatory part, right, upstream of it, which is going to regulate how this particular gene that is going to produce product. Okay, so in this diagram, the regulatory region, it is existing upstream of the gene, right, which is acting as a proto-oncogene. So now if any mutation happens in the coding part of the gene, so this is depicting a mutation, right? So what we can expect? Simply, definitely the product which will be produced, that will be showing altered function, right? Why? Because our coding sequence, that has been mutated. Simple. Okay, now the other way is that maybe the amplification of the gene occurs. So here we can see that there is just one copy of the gene, whereas here we could see that the amplification has occurred, right? So duplication of the gene. So what will happen? The excess of the protein that will be produced, increased synthesis of the encoded protein that will be seen. Now, the next case is probably where the translocations may happen. Means the DNA from some other location that may come here and it will get fused with the proto-oncogene, right? Now, in case the DNA regulatory sequence is being translocated from distant site. So now what we could see here, just have a look here. We had red region as the regulatory part. Now you could see that we are having a green region here, right? So it has been translocated from some foreign site. Let us say from some foreign site it has been translocated here and it has been fused with the proto-oncogene. So what would happen? The regulation which this red region was having over the coding part, that will get affected, and you will again find that there will be an excess production of the encoded protein. Or maybe the translocation, it will be affecting the coding part. So you could see this orange region, right? This is a protein coding gene which has been translocated from distant site, and because of that we could see that the phenotype of the protein that has been changed. So here what do we have? We have half of it in orange color, half of it in the purple color. Okay, so this is a fusion protein. It is again not normal, this is abnormal. Okay, so I hope this is clear how a proto-oncogene may get converted into oncogenes. Okay, now Both increased cell division and decreased apoptosis can contribute to tumorigenesis. This is really important, right? We have learned about the first part, how the increased cell division may happen, where we could see the role of oncogenes or the tumor suppressor genes, or maybe the defects which happen in the DNA repair system, right? The other part, the apoptosis, we are going to learn now. But let me tell you that both of them, right, they will get affected when the normal cell it undergo tumorigenesis. See, because the balance, right, the balance between these two events, that is going to keep a check over the cell division. So here, what do we have? We could see, let us suppose these are 4 cells, right, and now we could see here when they are dividing in a normal manner. Okay, so we could see that 4 of the other cells, or maybe the older cells which have been shown in the gray color, they are targeted for the apoptosis. Okay, so they'll be undergoing programmed cell death, and therein we'll find that the 4 cells, right, which are maintaining the homeostasis, right? Homeostasis means the normal physical, chemical condition of the body, right? So this is being kept in homeostasis because of the balance between the normal cell division event and the apoptosis event. However, if the cell division it shows an increase, so we can see increased cell division here, right? Normal apoptosis is there, but because the cell division it has increased, so again we could see that the tumor will be happening, right? Although the apoptosis is going in a normal way, but since the cells they are dividing in an abnormal way, so the tumor may appear. Or if in case we have normal cell division but decreased apoptosis, so we could see that apoptosis it has been significantly decreased, so it may again lead to tumor. Okay, so I hope this will help you to imagine that how a balance between these two events, that is maintaining the homeostasis in the cell and thus in the body. Okay, so from here now we need to move to our next part of the today's session, that is apoptosis. Okay, so apoptosis, definitely that is programmed cell death, right? So here we can say, if I just need to define apoptosis, so the demise of the cells by programmed cell death is marked by a well-defined sequence of morphological changes, which are collectively referred to as apoptosis. So whenever a cell will be undergoing apoptosis, it will be showing a well-defined sequence of morphological changes. You know, apoptosis actually, it has been taken from a Greek word, right? This is a Greek word and it means dropping off or falling off, like falling off the leaves. So from there the inspiration has been taken for using this word apoptosis, right, to mark the programmed cell uh, death event for a cell. Okay, so here the cell they undergo death but in a very neat and orderly manner. So what happens, you will find that the cell will shrink, right? And the nucleus that will shrink, the compaction of the chromatin will happen, the condensation of the cytoplasm will occur, okay? And then finally, the fragmentation will occur. So you will find that the entire cell, okay, the DNA, the cytoplasm, everything that is going to show fragmentation, okay? So that will get fragmented into small bodies which are known as apoptotic bodies. Okay, apoptotic bodies which are further engulfed by the phagocytic cells like macrophages. Right. So they will also be showing a blebbing. So what are blebbing? Blebbings, these are protrusions of the cell membranes. Okay, Sahiti, you have asked that until what time I'm going to take class. Okay, so I'm just going to finish this apoptosis, right? So maybe just next 10 to 15 minutes and we'll be done. Okay. So this part is important. I hope you have understood what all morphological changes they will be happening when a cell will be undergoing apoptosis, right? To simplify it, to put it in the simple words so that you can memorize it, remember the cell will shrink. and it will get fragmented into the apoptotic bodies, which are further engulfed by the phagocytic cells. And here we have another term, necrosis. If you remember, I had used this term earlier, okay, where I'd shown you a picture wherein the tumor cells, they were showing necrosis in the center. So what happens in the necrosis? Necrosis, that is also marked by the cell death, but this is not a programmed cell death. Okay, this may occur because of a trauma or a shock condition, and the cells, they will not be programmed for undergoing this kind of death. And here what will happen, the chromatin clumping, the swollen organelles, means the organelles, they will get swollen, and that will be disintegrating the cell, right? So disintegration of cell will happen, and all the intercellular contents, they will be spilled in the surroundings. Okay, so release of the intercellular contents occur, and that leads to the inflammation, right? But nothing like this you will find in apoptosis. This is a very neat process, neat and orderly process, right? The intercellular contents, they are not being released like this, okay, and due to that the inflammation will not be seen. Okay, so what happens here, DNA, that will also be undergoing fragmented, chromatin condensation, fragmentation of nucleus will happen, and like I've told you, the formation of apoptotic bodies. Okay, and these bodies, they will be further cleared by the phagocytic cells. Okay, now, so I hope you have understood that apoptosis, that means death, programmed cell death in a neat and orderly process. So what are the important characteristics associated with apoptosis? Overall shrinkage in volume of the cell, loss of adhesion to the neighboring cells, formation of blebs at the cell surface. So I had shown you what do we mean by blebs, protrusion of the membrane. Okay, dissection of the chromatin into small fragments, rapid engulfment of the corpse, whatever would be left, the apoptotic bodies and all, that will be cleared by the phagocytosis. Okay, Jaya, you have asked what is blebbing. So I show you once again. This is what do we mean by blebbing, right? These are blebs. So where we can see the cell membrane, it is protruding out. Okay, so these are known as blebs. So they will be further converting into the apoptotic bodies, right? So I hope that answers your query. Now let us move to our next term, which is really important in relation to apoptosis, and this is about knowing caspases. So caspases, these are a distinctive group of cysteine proteases that are activated at an early stage of apoptosis and are responsible for triggering most of the changes observed during cell death. Okay, now what are caspases? Caspases, these are the why they are known as caspases actually. They have key cysteine residues, and they cleave at a C terminal to the aspartate residues. So they are actually known as cysteine proteases. Okay, I repeat, they do have cysteine residue, and they cleave the proteins. They are known for cleaving the proteins, right, which will make a cell to undergo apoptosis. So I'll be explaining that on the next slide. Okay, so they cleave a protein at C terminal to the aspartate residues. Okay, now how caspases, they are involved in apoptosis. Okay, so they are in, firstly you need to know about the three categories, right, into which we can keep our caspases: initiator, executioner, and inflammatory. Again, let me tell you that it at this level you just need to know the names, right, and little bit about that, how the caspases, they are targeting the different proteins which will lead to apoptosis. But when you will be going further, right, when you will be learning the things in much more detail, then you are supposed to learn all the examples for the initiator, caspases, executioner, inflammatory, and even the detailed mechanisms how they will be contributing into apoptosis. Okay, but for now let me tell you that caspases, which can be kept into these three categories, they will be targeting The inhibitor of DNases, nuclear laminases, cytoskeletal proteins, Golgi matrix protein, or scramblers. So let me tell you that what is the significance of targeting these proteins by Caspases one by one, or the other components which we are finding here. Now, let me start with the nuclear lamins first. So we know that nuclear lamina, they support the nuclear envelope, right? And in the changes which are associated with apoptosis, we have seen that fragmentation of the material that will happen, fragmentation of DNA and everything. Okay, so the nuclear envelope that disappears. So there, the fragmentation of the nuclear envelope, disappearance of the nuclear envelope will happen, leading to the fragmentation of the nucleus. Okay, so there in the Caspases, they will targeting those nuclear lamins, right, which are supporting the lamina, which is further responsible for maintaining the integrity of the nuclear envelope. Okay, then they will be targeting the cytoskeletal proteins like actin, myosin, tubulin, vimentin. So they all are responsible for what? For maintaining the cell shape. Okay, so Caspases will be targeting this as well. So again, that will lead to the cytoskeletal disruption, cell fragmentation, membrane blebbing. Golgi matrix protein, so that will cause fragmentation of Golgi. Now this is very important. Scramblase. What is scramblase? Scramblase is a phospholipid. See, I have told you that the cell which is undergoing apoptosis, that will get converted into apoptotic bodies, which will be further engulfed by the phagocytic cells. Okay. So, how the phagocytic cell will identify, okay, that this is the apoptotic body and I have to clear. So, therein, there is the presence of phosphatidyl serine on the outer leaflet, right, of the membrane of this apoptotic body, uh, and the scramblase protein, scramblase phospholipid, this is a phospholipid, this will be helping in the translocation of phosphatidyl serine from the inner leaflet to the outer leaflet. Okay, I repeat, phosphatidyl serine, this is present on the inner leaflet. Okay, but scramblase will help to translocate phosphatidyl serine on the outer leaflet. So that will be recognized by the phagocytic cell as an eat me signal. Okay, and then they will be clearing out those apoptotic bodies by engulfing them. Then at first place we have inhibitor of DNase, ICAD. So what is CAD? First of all, CAD means Caspase activated DNase. Okay, Caspase activated DNA. So DNase that will be fragmenting the DNA, right? And I will use for the inhibitor. So there remains an inhibitor of this Caspase activated DNA. However, when that inhibitor will be cleaved by the Caspase, so these DNases, they will work and they will fragment the DNA. Did you get that? Okay, I repeat, Caspases will be targeting the inhibitor of the DNase, right? This is a DNase only, right? Caspase activated DNA, which will be causing the DNA to fragment. So when this inhibitor is cleaved, it is targeted by the caspase. So the DNA that will be making the DNA to get into fragments. Now moving to our next slide. And here, this is about caspase activation during apoptosis. Okay, so little bit idea I'm giving you that how the initiator or the executioner caspases, they are involved in causing or in making the cell to undergo apoptosis. Okay, so this is about the initiator caspases. So in initiator caspases, we have 2, 8, 9, 10. Okay, so here caspases 8 and 9, they have been shown. So they act as dimers, right? However, initially they will be in the form of inactive monomers, which are known as procaspases, right? So they do have a protease domain. They are, remember, they are proteases, and then they do have an adapter binding protein. Okay, so protease domain that remains at the carboxyl terminal region, whereas the protein interaction, this is the protein interaction, means they will be interacting with some proteins which will be bringing in the signals that these caspases, they turn to be active now. Okay, so it remains at the amino terminal. So now what happens? Let us suppose the apoptotic signal it has arrived, right? These are the proteins which are bringing in the apoptotic signal. So they will make these caspases to dimerize. Okay, they will get activated, right? The cleavage that will happen at the protease domain of the caspases, and therein some rearrangement will happen. Can you see this? Earlier it was like this, now it has been turned into this form. So the rearrangement that will occur, and it will make these caspases to turn active. Now these are active. So this is about the initiator caspases. Okay. Now this is about the executioner caspases. Okay, 3, 6, 7. These all are the executioner caspases. So they are also in the inactive form, inactive dimers. So they will be activated by the cleavages which will be made by the initiator caspases. They will turn active. Now this is the active form of the caspases, and then they will be causing cleavage of multiple substrates like we have learnt on the previous slide. These all could be the targets of the caspases. Okay, so they will be targeting all those proteins which will lead to finally the cell to undergo apoptosis. Okay, so I hope this is clear. Any doubt, let me know. This is how caspases they could be involved. Now you could ask that how the initiator caspases they will get the signal, right? What kind of signal they will get, from where they will get that, okay, now we have to get activated and then we have to further activate the executioner caspases. So there you need to know these two terms, right, which are related to the two pathways of the apoptosis. And like I've told you that you are supposed to learn the details of this pathway later, but not at this level. Okay, for now, the two pathways, intrinsic and extrinsic pathway, internal stimuli such as abnormalities in the DNA, that will be initiating with the intrinsic pathway. Okay, and intrinsic pathway of apoptosis that depends upon mitochondria. So this thing you need to note down. All right. Now, if we talk about the extrinsic pathway, so external stimuli such as certain cytokines or other factors which will be bringing in the signal, okay, cell surface death receptors, they activate the extrinsic pathway of apoptosis. Right? So I hope you have understood. So the signals they will come either from intrinsic or extrinsic, and the caspases they will get activated, and then they will be targeting the different proteins which will lead to apoptosis. Although these are two separate pathways, but remember there always remains a crosstalk between these two pathways. They could be related to each other. Okay, means external signals they can finally or eventually they may lead to the intrinsic pathway as well. Okay, so they could be connected, they could be related. So this is where we complete with our topics of the day, the cancer and apoptosis. So let us have a look over some questions to check whether you have been able to understand the concept or not. So this is a question here, and this is about that which of the following events will not usually lead to the transformation of a normal cell into a cancer cell? Yes. Which of the four options do you think will not usually lead to the transformation of a normal cell into a cancer cell? Yes, so I would like all of you to give drop your quick replies in the chat section. Gain of function of oncogenes, loss of function of tumor suppressor gene, gain of function of genes involved in nucleotide excision repair. This is one of the DNA repair mechanisms. Loss of function of pro-apoptosis related genes. What happened? Nobody is answering. Yes. Okay. So there I could see the answers popping up. All right. Jaya, you have given option 3. Nahid Fatima, option 4. Ajit, option 2. Okay. Pradumesh Gupta, option 3. So yes, option 3 is the correct answer. Yes, this should be option 3 only. Why it will be 1, 2 or 4? Gain of function of oncogenes, that we have understood, that if in case oncogenes they are gaining with some additional functions, right, which will make them overactive. So in that case, they will lead to cancer. Loss of function of tumor suppressor gene, that again we have understood. In case the tumor suppressor gene, they will lose their function, so that may again lead to cancer. Loss of function of pro-apoptosis related gene, so that will prevent the cell from undergoing apoptosis, that may again lead to cancer. Okay, so we are left with only option 3. Gain of function of genes involved in nucleotide excision repair. This is a repair mechanism. Okay, so DNA repair mechanism, anyways, this is not harmful to the integrity of the DNA, right, or the cell. So gain of function of a gene involved in repair, that will not lead to cancer. However, loss of function may lead to. Okay. Now, what do you mean by apoptosis? See, this is a question which has been asked previously. Now you have learned apoptosis. In fact, I have explained all the things in much detail. Okay, you know what all events they could be associated with apoptosis, right? The name of the proteins which are responsible for causing the apoptosis, how they can act, their categories, right? An overview over the mechanism, the two pathways. So I think now you know in detail, right? What do we mean by apoptosis? So this is really simple to answer this question here. Yes, let me know. Definitely. Okay, so this should be option 1 only. Okay, this was another question which was asked in the exam previously. What cancer day is observed every year on? So, do you have any idea on this? When we have World Cancer Day, anybody would like to try on this? Okay, so this is option 1. Okay, 4th Feb. All right. Now let us move to our next question. Which of the following is believed to be a key cause of the immortalization of cancer cells in many tumors? Yes. Which of the following is believed to be a key cause of the immortalization of cancer cells in many tumors? So let me answer over this question. This is shortening of telomeres. Remember, I had explained about the term replicative senescence. I told you that every normal cell, it will not be dividing in an indefinite manner. It will always be dividing in a definite manner because it will be showing replicative senescence, right? So beyond a particular limit, the cell will not be able to divide. The replication will not be able to happen. Why? Because the telomeres, they get shortened. Means there could be other reasons to to decide on that what will be the limit a cell will be following in terms of cellular division. However, so far telomere shortening, that is the best explanation, right? Which is going to determine that how many times the cell is going to divide. Okay, so shortening of telomeres, right? Because with every replication, remembers that telomeres, they get shortened. Okay, so when the telomerase enzyme, right? Telomerase enzyme is the enzyme which will be maintaining the telomeres, right? So telomeres, see, shortening of the telomeres that happen with every cell division. Okay, and this is the reason that cell has a particular uh, number of division it will be showing, right? And there we have a telomerase enzyme which is responsible for maintaining the length of telomeres. So if this telomerase enzyme that becomes functional in the somatic cells, so what would happen? The cell will not be following the replicative senescence, right? I hope I have been able to explain this. Shortening of telomere, that is responsible for determining the number of cellular divisions a cell will show. And the reactivation of the telomerase enzyme, that will be responsible for maintaining the length of the telomere. So if just imagine, if the telomere, they will always be maintained. So what would happen? The cell will not be showing the replicative senescence, and because of that, it will turn immortal. So this is the reason that we have option 3 as an answer here. Okay. So this was all about today's session. So I hope you found the session helpful. Okay, so I have this quote to share at the end. Everything is possible. The impossible just takes longer. So don't be impatient. Some things take time, experience, trial and error, and adjustment. So have a great day and think about how you cannot, why you can't. Okay, thank you all for participating in today's session. And don't forget to drop your comments, your likes. Okay, if you have found the session helpful, so don't forget to like the video. Fill the form, the link has been shared, and you can download the notes for today's session.